Hello, welcome to Cell Reproduction Lecture Video number 3. We are going to start on the topic of mitosis today. That's what we're going to talk about today. Mitosis, as you may recall from our previous lecture, has to do with the division of the nucleus and what is inside the nucleus that is dividing. Mainly, it is these things called chromosomes that are inside the nucleus. So this is a form of asexual reproduction that occurs in organisms that are trying to produce more body cells, for example. And another word for body cells is somatic cells. So if you notice, if you look at this picture, it starts with 4 chromosomes, an orange one, a blue one, a red one, and a purple one, 4 chromosomes. And by the end, it will eventually result in 2 new cells, and each cell will have 4 chromosomes in it. Yes, these are only half of one of these pieces, but they will eventually copy themselves and duplicate and eventually look like those original chromosomes. So mitosis, if you start with 4 chromosomes, you will end with 2 resulting daughter cells that also have 4 chromosomes inside of them, as shown here. Okay, so we're going to go through each part. This is the order in which they occur. Prophase, metaphase, anaphase, telophase. Now, interphase, which we spoke about in a previous lecture, is not part of mitosis. Interphase is not when the cell is dividing. The cell is preparing to divide and doing its normal stuff in interphase. But mitosis is when it's actually dividing. So these are the phases. Please make a turtle will help you remember the order. Please P make MA for anaphase and T turtle. Right there, that's the order in which they occur. So let's go through each one individually. Prophase is the first stage, and this is what it would look like for an actual cell under a microscope. So what happens in prophase? Well, it's preparing to divide in prophase. How does it prepare to divide? Well, the nuclear membrane holds all of the DNA inside of it. You can't divide what's inside of it if you have it inside of this membrane. Think of a membrane, the nuclear membrane, it's kind of like a bag. So the chromosomes are inside of a bag. Well, we need to break that bag. So the nucleolus that starts breaking down, the nuclear membrane starts breaking down and beginning to disappear. Okay, so the first thing that occurs is a lot of the nucleus begins to break down. Also, what happens is the DNA, which was chromatin before, and it was not visible when it was chromatin, it starts coiling and condensing into chromosomes that are now visible. Okay, so if you look in this picture here, if you look over here, you can't really see the chromosomes. They're not really visible. You just see a bunch of stuff. And over here, you're starting to see the chromosomes get tighter and coiling into actual visible chromosomes. Over here, they're chromatin. Over here, they're chromosomes. Over here, chromatin. Over here, they're starting to become chromosomes. Okay, what also occurs is these things called centrioles or centrosomes start to form, and centrioles are made out of microtubules, and what do? They help move things along on the inside of a cell. So if these chromosomes get divided, well, they're going to get moved by these fibers called spindle fibers that are also forming, and they begin moving towards opposite ends of each cell. Now, spindle fibers, those fibers I was just talking about, they'll eventually stretch from end to end. And why are they important? They're important because they are what move the chromosomes around in the cell. What are they made out of? You may remember in early unit, they're made out of these protein structures called microtubules. And these microtubules, they radiate away from a central area called a centrosome. All right, so these microtubules are going all the way across. They're also called spindle fibers, and they attach to the chromosomes. All right, this is called a mitotic spindle, or they call them spindle fibers. Right? The kinetochore fibers are the fibers that actually connect directly to the chromosomes. So here are kinetochore fibers. Whoops. The polar fibers are the ones that go from end to end. They're kind of like tracks so that they can slide along these tracks and move along. So the kinetochore fibers connect directly to the chromosomes at the centromere region, and the polar fibers go from end to end so that they serve as tracks for the chromosomes to move along. The second phase of mitosis is called metaphase. I tell my students, pretend the M for metaphase stands for middle. If you notice, the chromosomes line up down the middle. Now here's a picture showing you can see the mitotic spindle sort of on the edges here, and these are all the chromosomes lined up right down the middle. This is an actual microscopic picture of an animal cell in metaphase. Step 3, called anaphase. I tell you guys, pretend the A for anaphase stands for apart. What gets pulled apart in anaphase? Well, remember, these are sister chromatids. They're identical to each other. Two chromatids make up a chromosome. Well, the chromatids get pulled apart from each other so that they're identical copies of each other, so that one copy begins moving towards each side of the cell. Okay. How do they get pulled apart? The kinetochore spindle fibers are attached to the centromere, and they help pull them apart, basically. So anaphase apart, the chromosomes separate, they get pulled apart into chromatids. Even though these are half of an original chromosome, these chromatids are now considered full chromosomes. And this is showing you a picture. The spindle is going across, this is the centrosome, and it's pulling them towards a different direction. Fourth stage of mitosis is called telophase. In telophase, I tell you guys, it's kind of like the opposite of prophase. In prophase, the nucleolus, the nuclear membrane broke down. In telophase, they reform. The nucleolus, the nuclear membrane, the nuclear envelope reform, basically. Okay, they start to reform. Those fibers, those spindle fibers that are going across, they begin to disappear and break down. If you notice, the chromosomes are now located on each side or pole of the cell. All right? And the centrosomes that emanated from these centrioles, they start to break down. So, and finally, remember, in prophase, the chromosomes condensed and became, the DNA condensed and became chromosomes. In telophase, the chromosomes uncoil and become chromatin. They start loosening up. All right, and then you get a pinching effect happening. Okay, there's a pinching effect, and that pinching effect is called a furrow. Right. So here's the end of telophase. At the end of telophase, the cytoplasm has to divide, which means it has to divide up all the organelles that were in there, and you get a cleavage that forms, and a pinching, and then it pinches into two new daughter cells. Now, plant cells do not have this pinching effect. Since they have the cell wall that goes around there, it's a rigid structure. It does not allow them to pinch, so they do not pinch. So what happens with plant cells, they form what's called a cell plate right down the middle, and eventually that cell plate becomes the new cell wall. That's the only difference between an animal and a plant cell. Plant cells have to form a plate to form the cell wall. Animal cells do not have a cell wall, so they get a furrow, a pinch, and then they separate into two new daughter cells. Now, how is cell division controlled? Well, certain proteins that you produce, kind of like enzymes, they regulate and control the different steps of cell division. If something is not right, they do not allow the cell to go past that point. Now, when this process of regulating cell division, when something goes wrong, it leads to cancer. So this is going to eventually lead to why or how cancer forms. So a cell, during the cell cycle of a cell, the G1, the S, the G2, and finally it gets to the division point, the M and the cytokinesis, there are several checkpoints. Okay, so what are these checkpoints? Proteins. Okay, they allow it to continue on. They check what occurred before, and they allow the process to continue. If something is wrong, they will halt the cell cycle. So there is an M checkpoint that happens towards the end of mitosis to make sure everything else worked correctly. There is a G1 checkpoint that checks for cell size, nutrients, growth, DNA damage. Okay. There's a G2 checkpoint that gets it ready for division, for mitosis after that. Okay, so the G2 checkpoint says everything is good for DNA replication. Everything's good, after DNA replication. Everything is good after growth. So after the G2 checkpoint, you can enter mitosis. Okay, so let's talk about those checkpoints. First one, the G1 checkpoint. All right, so this one, it's going to control whether cell continues on. All right. The S phase comes after G1, and then during the S phase, in case you forgot, that's where the cell has to copy its DNA. If things aren't healthy, if the DNA is healthy, it will not allow it to continue on. If the cell isn't grown to the correct size, it will not allow it to continue on. So the G1 checkpoint stops it from copying its DNA basically and going to the S part. All right. If things are not favorable, and it can go into the cell rest cycle, the G0 phase. All right. If things don't go wrong, the cell, and by the way, at this checkpoint, certain cells, like I said, they will not be allowed to continue on. Certain cells will go into the G0 phase and maybe stay there forever from this checkpoint, okay? Then the G2 checkpoint. So I'm going to go back to show you the G2 checkpoint. So we just finished G1 checkpoint. Now we're all the way over here at G2 checkpoint. The G2 checkpoint is right before mitosis, in case you're forgetting. All right. At the G2 checkpoint, DNA enzymes, repair enzymes, they make sure that when it copied its DNA in the S phase, things did not go wrong. Bad DNA can lead to cancer. So this is how your cells can prevent cancer from happening. By not allowing these checkpoints will stop them from continuing on and becoming a bad cell. All right. If it passes this checkpoint, then it will enter mitosis. Okay. The mitosis checkpoint is the cell can only enter the next part, the growth phase, the G1 phase, if it passes and everything went okay during mitosis, then it will allow it to enter the G1. Now, what is cancer? Well, the proteins, we just talked about these proteins that regulate the growth and the division. Well, if a change in DNA occurs called a mutation, and it affects the genes that produce these proteins that are needed, well, that is one of the ways that cancer can form. So if those checkpoint proteins are not working correctly because your DNA has been damaged, and how does DNA get damaged? It gets changed due to a mutation. Then your cell will divide and grow uncontrollably, and that is what cancer is. All right? So cancer cells, they don't respond to those checkpoints normally. They don't do what they're supposed to do. They generally grow very rapidly. They suck nutrients from the cells and the organs around them, and they pretty much are, they suck the life out of the individual. So they take the nutrition that should be going to other cells and they use it to help the tumor grow rather than feeding the organs that may need those nutrients. Some mutations can also lead, there's another way cancer can form. Cancer can also form if people make too many growth promoting molecules, like specific hormones that help you grow. If your body makes too many of them, they can cause cell division to happen too rapidly, and then boom, that could be another reason cancer happens. So cancer happens when the proteins that regulate cell division are not working properly, or when you have a change in your DNA that leads to the overproduction of growth promoting factors like growth hormones and like insulin growth factor, for example, those can lead to cancer. All right. Now, the whole idea is you need cell division, and the cell cycle has to be controlled. When the control is lost, that is generally when cancer happens. And these are skin cancer, the most common types of skin cancer. I'm showing you this because skin cancer is one of the few cancers that we can see, therefore we should go get treated. A lot of times they start out as moles, and then those moles continue changing, mutations occur, and that mole can lead to cancer. Sometimes your doctors will remove the mole before it has a chance of changing and becoming a bad cell that can lead to cancer. So these are the very common types: melanoma, basal cell carcinoma, and squamous cell carcinoma. If you have an abnormal mole or an abnormal spot, it's not always a dark color like a mole. Sometimes they're pink and reddish in color. Sometimes they look like a scab that just doesn't heal. If you have any blemishes, you need to have them looked at by a doctor. We will stop there.